Okay, let's start back by looking at a first messenger and then to a second messenger. So as mentioned at the end of the last video, the ligand acts as a first messenger, and the important thing here, like epinephrine, is it's a protein molecule, and a protein can't cross through the cell membrane. Now, if this was a lipid-based molecule, well, then it could go right on through the cell membrane. So when we deal with protein hormones or messengers in the body that are proteins, they can't go through the cell membrane, so they're going to attach to our first messenger. So, they are our first messenger, and they're going to attach to a protein located in the cell membrane. Now, when we look at this, depending on it, you have your G proteins, you have your tyrosine kinases, and you have your ion channels. Those are all three different types of plasma membrane protein receptors. Now, when we look at like the G protein, for example, it's going to use what's called a second messenger. Now, cyclic AMP is one type of second messenger. There are other second messengers in our cells, and the main purpose of this is to pass the message on or relay the message. So while epinephrine was our first messenger, cyclic AMP is going to be our second. And cyclic AMP's job as a second messenger is to travel in the cell membrane, in the cytoplasm, and find the first protein kinase. Now, the first protein kinase is called PKA, protein kinase A, and it's inactive. When cyclic AMP activates it, that protein kinase goes through what's called a conformational change in shape. Now, because kinases' jobs are to transfer phosphates, what's going to happen is this kinase right here, this one, protein kinase A, is going to hydrolyze or use hydrolysis to break the bond and break that phosphate off of ATP, creating a DP and a phosphate. It will transfer a phosphate to PK2, protein kinase 2, basically phosphorylating it, and when it does that, that kinase is now in its active form. It also goes through a conformational change in shape. You see how it's changed shape. Now that protein kinase will then also transfer a phosphate group by taking a phosphate off of ATP and then phosphorylating protein kinase 3. That leads to a conformational change, and now it's active. Now the final step is going to be to activate a cell response. Now in this example, it's going to activate a protein. So at the end of the phosphorylation cascade, this right here is your phosphorylation cascade. Phosphorylation cascade. And it's going to activate a protein. Now this protein here, here is something called a transcription factor. Now if you remember from 9th grade, when we make proteins, we make messenger RNA, like mRNA. Now, at the AP level, we learned that in order to make messenger RNA, you have to turn on the genes. You have to turn them on, and so what we do is we use transcription factors to turn on transcription. Then we turn on our genes, we make the mRNA, we make the proteins. So here, by activating this protein, we've possibly activated transcription factors. Oops, that's a T. Now, you could also, maybe it's an enzyme. So maybe we've activated an enzyme, and so if that's the case, well then the enzyme might be, like let's say it's an enzyme that was activated, maybe it's the enzyme that's going to take a water and then break the bonds, the glycosidic linkages, and then release sugar into the bloodstream. So here if this was an enzyme doing hydrolysis, we can see how that would make sense. Because earlier, epinephrine, that we had as our first messenger, epinephrine, is adrenaline. And adrenaline is during our fight or flight response. So if you're scared and you have to fight off a tiger, well, you would need more sugar. You'd need an increase in blood sugar. So this makes perfect sense that the cell response is to increase your blood sugar levels so you can make the ATP to fight off that tiger. Okay, so my point is, there's going to be different cell responses. Right here, you can see, like, oops, sorry, wrong way. You have different cell responses that could happen, whether it's activating transcription factors or activating proteins within the cell or within the nucleus. That's going to be your final cell response. And throughout the course of the year, we'll see different examples of cellular responses. Okay, so let's go ahead and look at, well, how do we undo this? And so this phosphorylation cascade doesn't stay active. So the first protein kinase goes back to its inactive form. And now to undo what happened, we have an enzyme called a protein phosphatase. And this enzyme right here is going to go and it's going to basically break off that phosphate group, most likely by hydrolysis to break the bonds. When the protein phosphatase removes the phosphate, so you can see right here that phosphate has been removed, and this protein, PK2, is going to go through a conformational change in shape and go back to its inactive form. Here you have the protein phosphatase enzyme again, is going to go and do hydrolysis and break off that phosphate, and that protein kinase goes back to its inactive form. Oops. And so now this phosphorylation cascade is ready for the next cyclic AMP to come and activate and start the whole process all over again. These phosphates are now free in the cytoplasm to be joined to another ADP to make ATP in the future. And this is the basics of how our cells communicate using protein-based ligands. Good job, guys.